this opportunity to talk a little bit about this topic. Asthma is one of the focus of the research of my research group. Asthma is disease, a chronic inflammatory disease of airways with high incidence level, especially in developed countries. In Portugal, for example, around 10% of the population presents asthma. This level is higher in our countries like USA, UK, and others. And these numbers tends to increase on the next decade. And associated to its disease, there are high costs, direct and indirect costs, and also several problems associated with quality of life of the patients and also of their families. Today morning we have interesting talk about asthma in a different perspective, but there are some information about asthma symptoms and also pathophysiology of the disease, so I overcome this topic. And I want to highlight these aspects. There are the aspects central of my research, so it's the management of the asthma. What is important to do? So this is a so heterogeneous disease, and its control involves a lot of parameters, because also the genetics is important, the environmental conditions, and then a lot of parameters related to disease control, the therapy, all things related to the daily life of the patients. And I want to highlight some aspects very important: the early diagnostic, the knowledge of metabolic pathways related to the disease, and also the therapy follow-up, because there are a lot of therapies and combinations of different therapies used to asthma control. Right now there are novel specific immunotherapies against specific allergens, and it's very important to check the individual response. What is the most convenient therapy for each individual? To rationalize costs and improve the quality of life of the patient is always one of the highest focus. So to go further on this area, we designed a research that aims to join experts from different fields, from biochemistry, from different instrumental techniques, from metabolomics and bioinformatics, because we use commercial softwares, but we also developed several softwares with specific aims. Also a group from mechanical engineering, because we have developed a device to collect the alveolar fraction from breath. We also involved staff from immunoallergology and pediatric departments from 4 hospitals in Portugal. And the final objective of this project was to develop rapid and non-invasive tools that may be applied in clinical scenario to help to asthma control. In the first approach, our idea is to use metabolomic approach, as it allows a comprehensive study of the metabolism, to study in a deep way the metabolites produced by a biological system, and also to study the perturbations in the metabolism that allow us to study the different conditions, the disease status, or the effect of therapy among phenotypes, also among other relevant aspects. But to go there, it is important to establish a workflow. It is important to begin with the patient's treatment, and also to develop instrumental technology, also to look for the instrumental data cell processing in order to establish a pattern of biomarkers or a network of biomarkers that allow to extract relevant information. We decided to use breath in condensate and vapor phases, and also urine, because they may be starting in a non-invasive way, and they may provide information about the metabolism. This one more related to the respiratory system, but not only about the respiratory system, but in this one information can provide information about all the metabolism of each subject. And what type of methodology you can use? So if you can check metabolomics studies on the last years, we can observe that NMR is one of the biggest for important technology in this area, but also LCMS and GCMS. So we decided to use NMR, also established in our lab, and also to develop some improvements in gas chromatography, namely using two-dimensional gas chromatography, and finally also use some LCMS experiments. So we decided globally, we decided to define this type of workflow. So these three types of samples, you study NMR, we use NMR for urine, we don't have good signals for breath condensate. The signals, it's not enough to extract relevant information, and we use gas chromatography combined with mass spectrometry for all three types of samples. And then when you use gas chromatography, we also want to optimize and to select adequate extraction methodology, and then we use different types of data pre-processing and processing, depending if you want to look for specific metabolites or you want to targeted analysis, a comprehensive study of all metabolites detected. And also several aspects related with alignment, identification, to go further on the understanding of pathways related to this disease. One of the important aspects are related to the sampling, to define representative groups for control, for healthy group selected as control group, and also representative groups of the disease. But as asthma is a heterogeneous disease, so this group is very, for us, it's very big, as it included the active population, adults with different severity of the disease, and this is in stable conditions, in exacerbation. So this is a very big population. It is also important to collect information about individual and clinical data, also to allow to explain the differences in metabolism. For condensate breath, we use a conventional device, and for exhaust breath in vapor phase, we use a tabular sampling bags, but it is collected with a device that we developed in our lab, combining a CO2 sensor with a flow sensor, also to confirm that we have really a alveolar fraction and not a dead volume, this volume that is not significant to study perturbation that may occur at alveolar level. To select metabolites in combination with gas chromatography, we decided to use solid phase microextraction with this design. There are commercial available our designs. This is the most common. That in one step you can have the sampling, concentration, and extraction. And you can use it for the three types of samples. for liquid and also for vapor phase sample. There are commercially available different coating phases with different properties, polar, non-polar, that combine absorption, or other that are also some mixtures of coating phases, especially interesting for comprehensive studies, not for targeted studies. And what type of gas chromatography? So we can use different types of gas chromatography. This is one of the most common, but in this case we can quantify and also identify each compound related to each peak if we have the chemical standard. So, obviously in a metabolomic study we don't have standards to confirm all the instrumental signals. So if you have mass spectrometry, you can also have a mass spectrum and you can tentatively identify the metabolite. But using GC combined with the mass spectrometry and time of flight analyzer, we can go further on sensitivity, on resolution, chromatographic resolution, and also on spectral resolution, because we have a two-dimensional chromatogram and we can select appropriate columns to have specific regions. We can then define specific regions for compounds structurally related. This is particularly important for this type of studies because we don't have standards, we don't know exactly what is the metabolite, but we can check the chemical family. So if you can check the chemical families, we can go further on pathways. And what is the GC times GC? So we have two columns connected by a cryo modulator using liquid nitrogen. And for example, if you use a set of no polar with a polar column, what happens? So on the first column, we can separate the compounds according to their volatility, so clearly relate to the number of carbons. And then a small fraction or reinject on the second dimension, and you can separate the metabolites according to their volatility. So it's easy for us to know the number of carbons according to the position and the polarity. For example, we know that alkanes are here. And then you have the aromatic alkanes, and there the alkanes with a double bond, and so on, the alcohols, aldehydes, and so on. We can check the position of different metabolites according to their chemical structures. So basically, we have this type of workflow. So optimize all the parameters for the solid phase microextraction for different biofluids, and then you do the extraction, the analysis using this type of equipment. So this is also very, very important to look for the data treatment. We use, for example, univariate analysis or multivariate analysis, depending what you want to extract. We can also use unsupervised methodology, chemometrics, or supervised, and like PLSDA. And if you use supervised, it's important to validate. You use validation methodologies. For example, use ASCA when you want to check the effect of several parameters on the same subject. You also determine the VIP values. This is important to select the variables with higher importance in the projection. And it is very nice because we have a lot. We have for each sample we have hundreds of metabolites, and VIP values allow us to select the most important metabolites, reducing the time of analysis, and it's user-friendly technology, especially to apply it in a clinical scenario. Also, one of the biggest challenges are related to the peak alignment and peak identification, because it's a hard task, especially the peak identification. It's it's a really hard task, and you combine all of these parameters to tentatively identify, or in some cases when we have standards, to inject in standards and confirm the identification. We can, using this methodology, we can have low detection limits, micrograms per liter. Even if you have a trace compound, you can obtain a mass spectrum very close to the mass spectrum of the library. I think it doesn't work well. Here we have an example of the chromatogram from the different biofluids, and you can see that the chromatograms are quite different. Here you can select more non-polar compounds, and on these aqueous matrices we have more polar compounds. This information it's very complementary. It's quite complex. The figure may not show it. Here we have around 700 of metabolites, 400 and 300. So to reduce the complexity, sometimes what you do, we select specific diagnostics aims to reduce the complexity and the information and to highlight only some groups. For example, for us, these three groups are very important because three groups are related to stress, oxidative stress, and lipid peroxidation. That is a perturbation in metabolism that we identify in asthma. So using this pre-processing of the mass spectral data, we can increase selectivity and sensitivity. So now I can show some applications. Applications related, for example, using breath data, we can have a discrimination with also the validation, multi-carbon validation. We can have a discrimination between asthmatic and control group, and we also can follow the treatment effect in a child. We can also use VIP values to reduce all of this data to a small 9 metabolites that also allow us to predict the diagnostic the disease. We can also use this type of methodology, for example, to follow up the therapy. It's a specific therapy for a child, a teenager with severe asthma, and her doctor decide to start the therapy against mites. So the teenager on the first moment are here on the asthma group, and then after the treatment she is here. But she decided by herself to stop the therapy because it's not an easy therapy. Each 15 days it's necessary to go to the hospital. So she decided to stop the therapy, and she became to metabolic profile similar to asthmatic. And this difference on the metabolic profile are closely related to clinical symptoms. You also use it to study other, for example, relate to clinical parameters, for example, to eosinophils level, and also parameter related to lung function. But we can shift this correlation if the patients with index mass, with the body mass index higher than 50 who are excluded. So the obesity has also an effect on individual asthma response. And right now you are studying this effect, combining also NMR data. Here you can study the exacerbation of adult population in a stable and the same adults in a exacerbation condition, and you can check the increase of the metabolites related to oxidative stress and lipid peroxidation. Also, the NMR show that on exacerbation, compound metabolites related to oxidative stress are increasing and also related to PCA cycle. It is important to improve antioxidant therapies or diet therapies. Also, we have a lot of metabolites related to other disorders related to asthma. But to conclude, so we have used this type of workflow, and it's important to conclude that it's important to use the different biofluids and the different technologies because to construct asthma on its line, because the data are complementary. We can check information about all of these metabolic change related, highlighted on this figure, and we are using it in several scenarios. Right now, we are also using LCMS to establish the purine profile, as purine is also important signaling molecules in asthma disease. Finally, to thank all the financial support and all the team that participate on this study. For more information, you can check the website on my research group, and thank you for your attention.
Questions, Silvia?
Presentation. I would like to ask you if you do the NMR after purification or
no, you use NMR as indicating your workflow. You use NMR directly on urine sample by adding depurated water. By dilution, you have the urine. We do some dilution using depurated water and no more.
And you have a mixture of compounds there.
We analyze the urine directly. The extraction step is only to perform gas chromatography analysis.
I don't understand very well how.
No, the urine was analyzed directly by NMR. I also performed some dilution using filtrated water. And then you put urine in the tube and then in the equipment without any more handling.
To see how many compounds you have in the NMR of the urine?
So the urine, I don't have time to speak about it. Here we have the urine spectrum. And we want to know how many compounds.
If you are able, without the purification, if you are able to see
without any purification, we extract urine directly only after dilution.
And you have standards to compare or something?
Yeah, you have a lot of standards that we are used, and we also are working using the database of Booker in England. But without any previous purification, direct analysis of diluted urine.
A small question is for therapy. With these studies, you could predict how the patients are going to react to therapy, or you didn't think about it yet?
About it?
With these studies on the metabolism of the patients, you can predict how they are going to react to therapy and to metabolize the drugs, or you didn't think about it yet?
No, we studied the impact of the therapy on the metabolism.
Okay.
So if the doctor prescribes specific drug, we check the impact of this drug before and after the drug prescription.
Not the drug
Then if you want, you can speak a little bit more about this.
Silvia, I'm kind of interested in the reproducibility of the data. So if you take two breath samples as a replicate, how similar do they look when you expose them to this sort of analysis? If you take two breath samples from the same patient,
in vapor phase
how similar are they?
and in condensate. Is there a guess?
What is the similarity? How reproducible are the results? If you
asked about it. Now, we also check the intra-individual variety. We study the same individual through several months to check the variability. So we have variability within the day and within the days. But we have a pattern of the compounds that is specific for each individual.
But is it changing on a scale of minutes? Because it seems like people's metabolism at different times of the day.
Yeah, the metabolism is dynamic, but in the normalization of the data, we also use the normalization by total signal. So the absolute content is different. You have a pattern, and if you normalize the data for the total of the signal, this pattern is constant among the day in the days.
Thank you, Silvia. Okay, our penultimate speaker for today's session is Samara Silva de Souza, coming from the Federal University of Santa Catarina in Brazil. And the title of her talk is